Well, as the dust has settled after this year's ASCO 2025, we thought we'd take an opportunity to discuss some of the data updates in EGFR-mutated lung cancer. I'm here with Dr. Pasi, who's the director of the Chen-Huang EGFR Center at the Dana-Farber Cancer Institute. And I'm Julia Rotow, clinical director in the thoracic oncology program. And we thought we'd take you through our thoughts on some of the new data that's come out in this space. Dr. Jänne, thanks for joining me here today for this discussion.
My pleasure.
So first off, I thought we'd talk about some of the data in advanced metastatic EGFR-mutated lung cancer. And some of the interesting, I think, much awaited data we saw was in the ADC space. And I'm wondering if you might be able to take us through what caught your attention for ADCs in EGFR-mutated lung cancer this year.
So I thought this year we had some both exciting and maybe a little bit disappointing data in the ADC space. We saw that a TROP2 ADC, sacituzumab tirumotecan, or SAC-TMT, had a randomized trial called the OptiTROP-Lung03 trial versus docetaxel as second-line therapy for patients with advanced EGFR-mutant non-small cell lung cancer. And this trial demonstrated a significant improvement in both in progression-free survival favoring SAC-TMT, as well as an improvement in overall response rate. So this was an encouraging finding, and even a preliminary suggestion of an improvement in OS, but still the follow-up is short. So builds upon our previous experience collectively in TROP2 targeting antibody drug conjugates with datopotamab deruxtecan, which has also shown encouraging activity specifically in EGFR mutant subset of non-small cell lung cancer. So an exciting development there. I think the slightly disappointing development was the HER3 antibody drug conjugate patritumab deruxtecan, which had previously shown single agent activity in both phase 1 and phase 2 trials in patients with EGFR mutant lung cancer that had received EGFR inhibitors and chemotherapy. And at this year's ASCO, in a population of patients that had only received EGFR TKIs and not prior chemotherapy, we saw the results of the HERTHENA-Lung02 trial randomizing patients to platinum-based chemotherapy or patritumab deruxtecan. And while there was an improvement in a modest improvement in progression-free survival, there was no improvement in overall survival. And at the time of ASCO, we also learned that the sponsor, Daiichi Sankyo, had pulled the submission to the FDA for regulatory approval of HER3-DXd. So that was a little bit disappointing since this was an agent that had previously shown activity. But I think overall we're happy that there are potential ADC options emerging with TROP2 targeting ADCs for patients with EGFR mutant lung cancer. So Dr. Rotow, maybe I'll turn it over to you and talk about some of the MET combination studies that we saw at ASCO this year.
Yeah, and these were also interesting. And in some ways, the flip side of the ADC trials, which were more of an all-comer directed strategy at acquired resistance, we saw multiple different studies attempting to target MET at acquired EGFR TKI resistance. And of course, we know MET activation is one of the most commonly identifiable mechanisms of acquired resistance. So we actually saw two studies looking at adding MET TKI therapy, the SACHI study and the Savannah study, both randomized, both evaluating osimertinib combined with the MET TKI savolitinib at prior TKI resistance. And they showed some overlap in data. So the SACHI study compared to chemotherapy. It was an international study, run internationally, I should say, and showed improvements in progression-free survival. And same with the Savannah study. This was a combination versus savolitinib OC versus savolitinib plus placebo. And again, showed improvements in response rate and improvements in progression-free survival with continued TKI backbone. Now, these studies were interesting in that they differed in how they defined MET-driven resistance. One utilized FISH-based amplification as their measure in the SACHI study, and one used both MET-IHC and MET amplification as their biomarker. So speaks to one of the challenges when we're thinking about targeting MET in a clinical context. How will we understand what it means to truly be MET driven or MET positive as a clinically actionable biomarker in practice? But in this case, both produce similar PFS in the 8-month range versus 3 to 4 months with these very kind of slightly unique biomarker strategies. Now, interestingly, I think the Savannah study also got at a question that comes up a lot in the sense of when you get acquired resistance, you're targeting it in EGFR. This is often clonal and heterogeneous. So often we think the TKI backbone may be of use. And here, indeed, continuing the TKI did seem to offer benefit compared to MET inhibitor alone, which makes sense and has been shown before as well, for example, in the tepotinib osimertinib data in prior studies. Now, one third very interesting MET-directed study that we saw data from here this year at ASCO was the data for ABBV-400, which is a novel MET-directed ADC. And this, though, in earlier phase development, not a randomized trial, showed a very interesting response rate of 63%. And this was as monotherapy. Again, that's unusual in the MET-directed area. We've seen prior efforts with MET ADCs have required a MET biomarker for high-level kind of MET overexpression or high-level MET activation to show activity. So this was interesting, absent that MET biomarker getting response rates. So I think that's certainly something maybe unexpected in the MET space in EGFR. Now, I think one other topic we've been looking at in EGFR space has been the role of EGFR-directed therapies in locally advanced or early stage disease. And I thought I'd bring up something that caught my eye at ASCO, of course, which was also very much awaited, which was the NeoDora clinical trial data, which was a randomized study looking at integrating EGFR TKI therapy into the neoadjuvant setting. We've been doing adjuvant TKI therapy for some time in this space, but the question was, like immunotherapy, should this be moving into more of a perioperative or neoadjuvant strategy? And I will say, I think, Dr. Jänne, you may agree, this is early days on that study. We don't yet have access to mature survival data. So we don't have mature EFS or mature overall survival, which are our gold standards for any perioperative or a strategy. We did, however, get a look at ASCO this year at the pathologic response rate data. And I think it might have been expected based on prior studies and single arm trials. The complete response rate was quite low. We did see pathologic major responses in about 20% of patients, either on osimertinib alone or osimertinib plus chemotherapy, with very, very low major response rates to chemotherapy alone. So then the question is, and some trend towards better EFS, I think, with upfront combination with TKI alone, though again, very immature. So the question is what to do with this in practice. I think many of us would say that it's not yet time to integrate this kind of strategy into all of our patients who are getting resectable upfront EGFR-mutant lung cancer. But I think there could be an argument made for those patients who do require neoadjuvant therapy. If nothing else, the study tells me that I wouldn't want to offer neoadjuvant chemo alone, and I'd probably consider osimertinib plus chemotherapy in that setting. So the borderline resectable or the N2 positive patient, for example. I think we're awaiting longer-term mature survival data. Now, turning attention, one other topic that I think we wanted to cover here today were, of course, the uncommon EGFR mutations. So outside of the classical EGFR drivers, we saw some updates for EGFR exon 20 inhibition. I think we saw a zipalertinib update this year. And Pasi, I think, Dr. Jänne, I think you were going to bring that one up.
Yes, no, absolutely. At the moment, we only have amivantamab approved by itself or as a frontline therapy with chemotherapy for advanced EGFR exon 20 mutant non-small cell lung cancer. But there are many EGFR exon targeting kinase inhibitors that are being that are undergoing clinical development. And this year we saw an update on zipalertinib in both patients who had prior chemotherapy without any other exon 20 targeted therapy, or those who had prior amivantamab. And we saw very nicely their response rates were about 30% in the patients that had received only response rates were about 35% in the entire patient population, and a little bit higher in those who had not received any exon 20 targeted therapy, and a little bit lower in those who had received prior amivantamab or other exon 20 targeted therapy. but about 25%. And in general, the response is also extended into patients who had brain metastasis, which was wonderful to see. This agent is currently undergoing a phase 3 clinical evaluation in combination with chemotherapy, compared to chemotherapy in combination with chemotherapy. So it's exciting to see encouraging activity of these new kinase inhibitors as we need more therapeutic options for patients with exon 20 insertion mutations. And with that, Dr. Rotow, I'll turn it back to you.
Yeah, I completely agree. So thanks for chatting with me today. We'll continue to bring you some brief updates, hopefully after some conferences this fall, as well as we continue to watch see the field evolve. So Dr. Jänne, thanks for joining me here today and wishing everyone a great day.